Hi viewers, this is Abdul Rahman and welcome back to Students Diary. Today we will discuss yet another important topic of the carbohydrate metabolism, and this time it is about the pentose phosphate pathway, or we know it as Hexose Monophosphate Shunt pathway. Pentose phosphate pathway is a kind of alternative pathway of glucose oxidation, and this time this pathway is not meant for ATP production. Rest of the pathways which we have discussed about in the carbohydrate metabolism are mostly utilized for the ATP production or for the regeneration of the ATP, but the purpose of this pathway is not the ATP production, but rather it give you a molecule of nicotinamide dinucleotide phosphate, and it is the most important product of this pathway because it has role in certain anabolic processes. Similarly, it gives you the ribose 5-phosphate, which is again the very important structural component of the nucleotides and nucleic acid. We know that ribose is the part of the RNA, while deoxyribose is the part of the DNA. So this pathway, which is the pentose phosphate pathway, is alternative branch of glycolysis, and this give you the sugar molecules, which is ribose, and that help in making up DNA and RNA. So when we see that glucose is taken up by the cell, and then most of the time it is taken into the glycolysis process, and on the way it give you the glucose 6-phosphate, and then multiple of enzymatic reaction it lead to the Krebs cycle, and the Krebs cycle lead to the electron transport chain. So all these processes gives you energy generation in the form of NADH and ATPs, while the pentose phosphate pathway originate from the glucose 6-phosphate. We know that glucose 6-phosphate is a kind of intermediate that could either way go to the gluconeogenesis, to the pentose phosphate pathway. It can go to the glycogenesis. So this is kind of intermediate molecule. So when body demands, that can move toward the pentose phosphate pathway. So this pentose phosphate pathway are mostly operated in the cytoplasm. Not mostly, it is in fact it is operated in the cytoplasm, and it has the two phases. One is the oxidative phase, which give you a ribulose phosphate on in the process, and similarly it give you two molecule of NADPH. While the non-oxidative phase, it has three outcome. One is it give you the ribose 5-phosphate on the way, it give you fructose 6-phosphate, and it give you glyceraldehyde 3-phosphate molecule. So ribose 5-phosphate, you all know that ribose has an important role. It is a 5-carbon molecule in RNA and DNA, but the fructose 6-phosphates, you are familiar with that. It is also kind of intermediate in the glycolysis pathway, and glyceraldehyde 3-phosphate is also come in a splitting phase of the glycolysis. We will see that how we can utilize this one in the pentose phosphate pathway. Now first come to the oxidative phase of the pentose phosphate pathway, and it is consist of three reactions. We will see it in this diagram, that in the first reaction the glucose 6-phosphate is converted with the help of glucose 6-phosphate dehydrogenase into glucose phosphogluconolactone molecule. Again, it is a 6-carbon molecule, but in the process it gives you NADPH molecule, which is very much important for the cells because it have an antioxidant role and other functions which we will discuss. And similarly, the G6PD, or we call it as glucose 6-phosphate dehydrogenase, is also very important diagnostically because we had a condition of G6PD deficiency. We will also discuss this in detail. So keep this one in mind. We will discuss this one in the last slides. Then in the next reaction, that 6-phosphogluconolactone is with the help of lactonase enzyme, and with the addition of the water molecule, it gives you 6-phosphogluconate, and that 6-phosphogluconate again with another dehydrogenase enzyme, and this time is 6-phosphogluconate dehydrogenase. It will give you another NADPH molecules with the removal of the carbon molecule. So with that removal of the carbon molecule, that molecule will be converted into ribulose 5-phosphate, and that will also give you the second NADPH molecule over here. Remember, we had discussed earlier that wherever the dehydrogenase enzyme is involved, it most of the time give you NADPH molecules or NADH molecules. While if there are any kinase enzyme involved, there are certain phosphorylation or dephosphorylation happen and the utilization or ATP production takes place. So this was about the oxidative phase, which started on from glucose 6-phosphate and end up in ribulose 5-phosphate. So it gives you two important molecule. One is ribulose 5-phosphate and the other one is two molecule of NADPH. The ribulose 5-phosphate will further lead to the non-oxidative phase, which we will discuss. But first we will see the utilization of NADPH and the importance of the NADPH, which has been generated in the oxidative phase for us. Oxidative, the cells in our body, most of them are required high amount of NADPH reserves, which come through this pentose phosphate pathway. So the utilization of the NADPH is for the reductive purposes, and most of these reductive processes are for lipid biosynthesis. We know that most of the hormones in our bodies are steroid in nature. Importantly, the fertility hormones are lipids or steroid in nature. So for that production, NADPH is also has an important role, and NADPH also has a very powerful antioxidant role. As I have told you earlier, that it combating the free radicals. So this is the kind of force which neutralize the effect of the free radicals, which we called as oxygen reactive species, ROS, reactive oxygen species, or RNS, reactive nitrogen species, in the form of hydrogen peroxide, which are generated in our body through normal processes. Oxidative phosphorylation process may lead to certain production of free radicals, but that will be neutralized by the NADPH, which is produced through this pentose phosphate pathway. One thing is more important, that free radicals in old age, when we have a weaker immune system and when we had less amount of this antioxidant production in our bodies or in our cells, these free radicals can lead to aging process because that will damage the tissues or cells. That may lead to different condition like cataract development in your eyes and other tissue damage which lead to the aging process. So this is one of the target for the modern medicines, to neutralize these free radicals more effectively in aged individuals, so that we can reverse or delay the processes of aging in that individual. Secondly, NADPH is very much important for red blood cells that help in iron to reduce form and it increase the affinity of the hemoglobin with the oxygen. More importantly, that NADPH reduce the glutathione, which help in combating that free radicals in the red blood cell. So it help in that to maintain that glutathione in reduced state. So that help in to preserve the integrity of the red blood cells. So maintain that shape of the red cell membrane with the help of that reduced glutathione level, and of course that reduction of the glutathione is required NADPH level in the red blood cells. Similarly, that NADPH has role in the phagocytosis process in the white blood cells. You know that white blood cells are kind of our immune force, and that the tool they are utilizing for engulfing the bacteria or destroying the bacteria, it required the NADPH, and that NADPH come from the pentose phosphate pathway. And another important thing of the NADPH is that its high concentration is very much important for the lens in our eyes, and it help in to preserve the transparency of the lenses. If NADPH level will be low in our eyes, that will may result to cataract formation or other damage to the lens. So thanks to this NADPH level, that it keep cleaning and keep preserving the transparencies of our lenses. The different pathways which require, or the processes which require the NADPH. We already mentioned that most of the fatty acid biosynthesis, cholesterol biosynthesis, neurotransmitter, nucleotide biosynthesis, all these required NADPH, and certain detoxification processes which require a reduced form of the glutathione, and similarly cytochrome P450 monooxygenase enzyme, which has the detoxification role, also require the NADPH level. And the tissue which basically contain the active pentose phosphate pathway that include most of the adrenal gland, liver, testes, adipose tissues, ovary, and mammary gland. All these tissues are mostly involved in steroid or fatty acid synthesis. Apart from red blood cells, which we had studied that it maintain the reduced glutathione level, which help in the membrane maintaining the shape of the red blood cells by maintaining the structure of the red blood cell membrane. Now come to the non-oxidative phase. We have mentioned that oxidative phase will end up on ribulose 5-phosphate, and from now here onward the ribulose 5-phosphate will come into the non-oxidative phase, and ribulose 5-phosphate will be converted into ribose 5-phosphate with the help of ribulose phosphate isomerase enzyme, or another way can happen that xylose 5-phosphate will be formed by ribulose phosphate isomerase. So that either way it can give you xylose 5-phosphate, or it can give you ribose 5-phosphate from the ribulose phosphate molecule. While the remaining of the reactions of the pathway, these will be kind of interconversion reaction. Enzyme transketolase or transaldolase will be involved, and that will result in the interconversion of these molecules and of the trioses, pentoses, and hexoses. So for these interconversion reaction and for these transketolases or transaldolase enzyme, TPP, that is thiamine pyrophosphate, will act as a coenzyme. We know that thiamine pyrophosphate is a vitamin B1, and the function of this transketolase enzyme is that to transfer 2 carbon units from the ketosis to aldoses. This process will explain this kind of flow diagram will further give you the information. This is the glucose 6-phosphate molecules, and here the 6th molecule of the glucose 6-phosphate molecule went into the oxidative phase of the pentose phosphate pathway. We will not go into detail as we have already studied the oxidative phase. So it give you the ribulose 5-phosphate, 6th molecule of the ribulose 5-phosphate molecule. Now this 6th molecule of ribulose 5-phosphate molecule will go either into with the help of ribulose 5-phosphate ketoisomerase enzyme give you 2 molecule of ribose 5-phosphate, or it will give you 4 molecule of xylose 5-phosphate molecule, and this time the enzyme will be ribulose 5-phosphate epimerase. So with the help of then interconversions of these 2 molecules, that will, with the help of the transketolase enzyme and the coenzyme which is involved is TPP molecule, which is vitamin B1, which will then it will give you 2 molecule of glyceraldehyde 3-phosphate. You will be familiar with this molecule. It is a 3 carbon molecule, so it will give you these 3 carbon molecules, while on the other hand it will give you 2 7 carbon molecule, which is pseudoheptulose 7-phosphate molecule. Again, interconversion between these two will take place, and this time the enzyme transaldolase will involve, and this time again it will give you 2 erythrose 4-phosphate molecule, and this erythrose, you know, is 4 carbon molecule, while on the other hand it will give you a fructose 6-phosphate molecule. This time it will give you 2 molecule of fructose 6-phosphate. So you know that 2 fructose 6-phosphate molecule can be easily converted back into glucose 6-phosphate molecule. On the way, that 2 molecule of the xylose 5-phosphate, which has been converted into these 2 molecules, the 2 of them will combine with some of the carbon molecule of the erythrose 4-phosphate molecule with the help of again transketolase and coenzyme TPP. It will give you glyceraldehyde 3-phosphate 2 molecule, and on the way, this glyceraldehyde 3-phosphate 2 molecule when combined will give you, because this is 3 carbon molecule, when this 3 carbon molecule will combine together, it will give you fructose 6-phosphate molecule. So overall, if I summarize you this whole process, some of the contribution of the carbon molecule from the xylose 5-phosphate molecule will give you fructose 6-phosphate directly with the combination of erythrose 4-phosphate molecule, and some of these erythrose phosphate directly will convert it into glyceraldehyde 3-phosphate molecule. So overall, 2 molecule of fructose phosphate will come here from here, and it will give you 2 from here. 1 molecule will be given from the glyceraldehyde 3-phosphate molecule, and from here 2 will come. So total 5 fructose 6-phosphate molecules will be regenerated. So and that will be again converted into glucose 6 phosphate, which will give you 5 molecule of glucose 6 phosphate molecule. So the story which has begun in oxidative phase, where 6 molecule of glucose 6 phosphate with the help of glucose 6 phosphate dehydrogenase, the end product of the first oxidative phase was ribulose 5 phosphate, and that 6 molecule of ribulose 5 phosphate goes through non-oxidative phase of these all reaction. It give you, regenerate you 6 molecule of glucose 6 phosphate again, but one molecule in the whole process is utilized in that oxidation process. So what was the purpose of this whole pentose phosphate pathway? The purpose was not about the generation of the ATP this time, but the purpose was to give you the ribose phosphate 5 phosphate on the way, and give you ribulose 5 phosphate, and it give you NADPH level, which is very much important for the different functions of the cells. Now comes to the glucose 6 phosphate dehydrogenase deficiency, which is the first reaction of the oxidative phase. If there is a deficiency of this enzyme, that could lead to hemolytic anemia. Anemia, you know, is the reduction of the red blood cells and hemoglobin level in a person. So this G6PD deficiency is an inherited sex-linked X trait or recessive disorder. So it means that it is X-linked, so it will be predominant in male because the normal X chromosomes will not be present in the male because there will be Y chromosomes, while the X chromosomes which has been present will be a mutant one. In that case, if he has that mutation in it, so that will lead to the predominant character of this disease in male as compared to female. While in female, most of the part, if one of the X chromosomes that contain the mutated copy of the glucose 6 phosphate dehydrogenase, that female will be carrier for that, because the female contain in another X chromosomes which are normal, and that will suppress the effect of this mutant gene. However, if the female receive both the mutant copy, one from their mother and other is from their father, in that case they will receive the double mutant copy. In that case, then they will not be carrier, but they will show the symptoms, and they will have that symptoms of the G6PD deficiency. We know that if there is a deficiency of G6PD, it will not lead to the oxidative phase, so no NADPH production will take place, and that will result in the effects on the cells of the individual, but these effects will be more predominant in the red blood cells. Remember, we have mentioned earlier that NADPH has a role to maintain the integrity of the red blood cell membrane and its shape. So if they are not present, and that will result in severe effect and hemolysis of the red blood cells, because hexose monophosphate shunt is the only means of providing NADPH in erythrocytes. Other tissues might have some other means of production of the NADPH, but hexose monophosphate shunt pathway is the only pathway in the red blood cells which give you the NADPH level. So that decreased activity of G6PD impair the synthesis of NADPH in the red blood cells. So this result in accumulation of the methemoglobins instead of oxyhemoglobin, and peroxide in erythrocyte lead to the hemolysis. So that will result in the more amount of free radicals in it that will disrupt the structure of the cell membrane, and it will result in the hemolysis. But the conditions can severe in that person, get worse in that person who are deficient of G6PD, if he got some severe infection or he use some kind of oxidant drugs. So the person which are G6PD deficient will be asked to avoid oxidant drugs, and similarly ingestion of the fava beans, because the person, if he eats the fava beans, it will result in the production of the oxidant molecules in his red blood cells. So there will be no NADPH or reduced glutathione to counteract that. So that will result into the hemolytic jaundice. We know there are 3 types of jaundice. One is pre-hepatic, other one is hepatic, and third one is the post-hepatic. Pre-hepatic is due to the red blood cells production, and one of the causes glucose 6 phosphate dehydrogenase deficiency, while other could be antigen antibodies reaction or immune response. If you give someone a false wrong blood group, it transfused wrong blood to someone, so that could also lead to the hemolytic jaundice, or we call it as pre-hepatic jaundice. The other one is hepatic jaundice, which may be due to certain kind of liver damages, as we had seen in hepatitis C and hepatitis B. While third one is the post-hepatic, which is due to some kind of blockage in the gallbladder or somewhere else due to stone formation. So that could, but in that case, that will be hemolytic jaundice, or we called as pre-hepatic jaundice if they are as glucose 6-phosphate deficiency. So sometimes this glucose 6-phosphate dehydrogenase deficiency could be a blessing in disguise for the individual, for those person where the malaria are very much common. I'm talking about the African population because the malarial parasites are totally dependent on Hexose Monophosphate Shunt pathway and reduced glutathione for their optimum growth in red blood cells. These parasites of the malaria are basically feeds on the red blood cells. They grow over there, they recycle over there. So if the red blood cells are not proper, have not proper shapes, or they are prone to rupture already because of these deficiency of the G6PD, and there are no NADPH, so there will be less chance for the parasites to grow in the red blood cells and multiply over there. So those person or those areas we have seen in Africa, which has both the genetic deficiency of G6PD and prevalence of the malaria, so these populations are resistant to malaria. So they are kind of blessing for disguise for them, because the red blood cells are deficient of NADPH, and that there is no chance for the malarial parasites to multiply and increase their parasite level in the red blood cells, which can lead to the symptoms of the malaria, which, so in that case it will not happen. So we call that as a natural selection. That is a positive mutation for that population, because that mutations help them in some way to avoid the malarial disease, because that malarial disease at some time was very dangerous, because there was no such treatment available for them, or even nowadays also it is a kind of second or third leading cause in the African countries, because malarias are common. So those people which are deficient of G6PD, which has that gene, are basically resistant to this condition. So this is kind of adaptability of the peoples living in the malaria infected region of the world. So this was all about the pentose phosphate pathway. Hope you will have get some knowledge from it. We will see you in the next lecture. Till then, Allah Hafiz, and don't forget to subscribe my YouTube channel.